Okay, so welcome to the last session before the poster session. We have 8 short talks, which are quite interesting, so I'm very excited to chair this session for you. As a reminder, speakers will have 10 minutes to present and more or less 2 minutes for questions. When you see me standing up, that means that you have 1 minute left, okay? So it's time to wrap everything up. So let's begin with the first speaker of this session, which is Jacopo Ronchi, which will present an integrative framework for miRNA-mRNA analysis.
Okay, so hello everybody. Today I'm going to present MIRit, which is an integrative framework for integrative microRNA and mRNA analysis. So first of all, I'm going to give you a brief introduction to the challenges of current microRNA research. The first issue is that the identification of differentially expressed microRNAs per se is not a sufficient condition to functionally evaluate their roles. And the main reason is that microRNA target interactions are ambiguous across different cell types. Therefore, integrative analysis can be a possible solution for evaluating the effect of individual microRNAs on their target genes. Nevertheless, current methodologies are quite tricky, let's say. For example, the combination of multiple prediction databases severely increases the number of interactions, and this is a problem for multiple testing. But also, they inflate the rates of false positives. And also, the different usage of prediction algorithms make reproducibility a big problem. Furthermore, the selection of statistical tests for the integration of microRNA and mRNA data is not always appropriate. For example, Pearson correlation is probably the most widely employed approach, but its assumptions are not always appropriate. Furthermore, simple correlations are not suitable for complex experimental designs, and also they may be subjected to Simpson's paradox. And finally, some of the methods are not lack formal validity. For example, lasso regression, which sometimes is used, is not adequate when the sample size is lower. And last but not least, there is a complete lack of suitable approaches for impaired datasets, and this heavily limits the availability of microRNA datasets and the integration with single-cell technologies. To address these problems, we developed MIRit, which is a unified framework for handling integrative microRNA and mRNA analysis from an all-in-one perspective. This is a diagram that shows how the MIRit framework is made. Now we're going to be a little bit closer. So first of all, differential expression analysis is carried out separately for microRNA and genes. Then MIRit retrieves putative targets by combining validated interactions and MIR-deep predictions, which is an ensemble method that combines different prediction algorithms by scoring them, and this leads to a reduced number of false positives and also a reduced number of meaningful interactions. Then for paired datasets, a partial correlation analysis using Spearman's correlation coefficient is used. This enables the use of complex experimental designs, and also, this is extremely important, to capture nonlinear relationships that are typical of microRNA target relationships. On the other hand, for unpaired datasets, association tests like Fisher's exit test or Bosch-Lewis exit test or rotation gene set testing can be used for unpaired datasets. Finally, MIRit provides functional enrichment of integrative targets, integrated targets, then deconstruction of a comprehensive network, and also extensive visualization options. So now let's see how we quantitatively compared the statistical tests involved in MIRit against the commonly used ones. So to do so, we have simulated 1,000 paired datasets, and differential expression has been applied on a subset of microRNAs, and different genes had different probabilities of being targeted, thereby mimicking the existence of hub targets. And also, different miRNAs had different, variable repression efficiencies, and their magnitudes were kept in a modest range to mimic the real microRNA data. And most importantly, the net perturbation of each target gene depended on the combination of multiple microRNAs, and this also considers the effects of thresholding and saturation, which are well described phenomena of microRNA target relationships. And lastly, random variation was added as well to simulate biological and technical noise. So let's take a look at the results. We compared we have compared the precision and the F1 score of Fisher-Zexek test as a representative of unpaired tests. Partial Pearson correlation, partial Spearman correlation, simple Spearman correlation, and simple Spearman correlation. So as we can see from the results, partial correlation is much more precise than simple correlation, and also we can see that Spearman's correlation is more effective than Pearson's correlation. But this is not the only thing that I want to show you. I want also to show you that even though Fisher-Zexek test is less effective than the other methods, its precision is still acceptable, and which is quite important for unpaired datasets when basically you have no choice. This is also seen by looking at the area under the precision recall curve. We see that partial correlation performs better than a simple correlation. And also we see that in both cases, Spearman correlation works better. So let's briefly look to case studies where we applied MIRit on real-world microRNA and mRNA data. First, we used it in a paired miRNA-mRNA dataset in clear cell renal cell carcinoma, where the expression of microRNAs and mRNAs in 17 patients were profiled using microarray technology. After exploring transcriptional variability and differential expression analysis, we used partial Spearman's correlation to unveil anti-correlated targets. And for example, we see here 3 examples of anti-correlated pairs where non-linear trends are evident. And also the integrated targets were depleted of multiple genes that are involved in the normal functioning of the human kidney, including ion absorption, which is a hallmark of renal carcinoma. And then we also applied MIRit on an unpaired datasets in Alzheimer's disease. And also in this case, we used Boschloo-Zexek test to identify significant association between 18 differentially expressed microRNAs and 940 target genes. And also in this case, when we performed the functional enrichment of these target genes, we have seen an important depletion of many brain function that unfortunately are lost in this pathology. So I'm going to wrap it up. So first of all, MIRit offers a comprehensive and all-in-one framework for both paired and unpaired datasets. partial coordination analysis reduces spurious associations that are probably caused by the group structure rather than real microRNA target influences. Then Spearman's correlation enhanced the capture of non-linear microRNA target relationships that are really common of microRNA target nature. And finally, I want to thank Professor Foti and all the collaborators from the University of Milano Bicocca, the organizer of this wonderful conference, and also I want to thank, most importantly, you all for the attention.
Okay, any questions for Jacopo? Well, maybe I can break the ice a little bit. I have a very naive question because I don't know about the field of microRNAs. But how do you exactly link microRNAs to their target mRNA?
Okay, basically there are several prediction algorithms, and also there are experimental databases where there are some molecular biology assays are used to functionally evaluate if the blockade of one microRNA have an effect on its target gene. The problem is that prediction algorithms are incredibly inaccurate, and they lead to a very high percentage of false discoveries. And we used MIRDIP, which is a great tool, actually, because it scores different prediction metrics from across different prediction databases, and so it retrieves a number of predicted targets that are much more relevant to the microRNAs in question. So,
nice. Any other question? No? Okay. Thank you, Jacopo. Okay, so the next speaker is Najla Abassi, and she will present One Class to Rule It All, DeeDeeExperiment for managing and exploring omics analysis results.
Hi. Okay. Can you please make it full screen? Okay, cool. Welcome, everyone. Hopefully you can see the one ring, the ring that's meant to rule them all. And in our universe, this ring is called DeeDeeExperiment. So usually, imagine that you have an omics data set. It can be a transcriptomic, metabolomics, whatever. Usually this kind of data is stored in a structure based or similar to the summarized experiment object. What you're gonna do with your with this data set is to perform you're gonna perform differential expression analysis, and then you're gonna do run your analysis on different contrasts, probably also using different frameworks. Then you're gonna do your functional enrichment analysis, which I call FEA, and honestly, in my humble experience, it has been an ugly situation. And if you speak Spanish, probably you get an extra joke in here, because FEA is ugly in Spanish. So basically, we have all these tables. We have too many tables. And the problem is also amplified in the context of single-cell data, where the pseudobulk analysis generates multiple tables across multiple cell types and multiple contrasts. So soon the situation can be overwhelmed with all these tables from the DEAs and FEAs. The problems, The problem is now we generated results that are difficult not only to manage and explore, but also to understand, contextualize, and reproduce. And to solve this issue, we created DeeDeeExperiment, a new S4 object to structure and store DEAs and FEAs all in one place. So basically, in a nutshell, yes, I didn't say that DeeDeeExperiment inherits from the single-cell experiment object. So DeeDeeExperiment brings in all the slots that comes with a single-cell experiment object, but also adds 2 new slots. The DEA slot where the DEA results table will go in with some extra metadata. And also for each feature, we store also the results in the metadata in a compact manner. And the second slot is the FEA slot in green, where we store the FEA results table in a structured manner along with some metadata. So how do we create a DeeDeeExperiment object? We use the constructor function DeeDeeExperiment. We pass a single-cell experiment object or something similar, and we pass a named list of contrasts. In this example, we pass 2 contrasts here, and then we create an object of class DeeDeeExperiment, and when we look at the slot there, we can see that we added 2 contrasts. Hello. So basically, the supported DEA results that you can insert in a DeeDeeExperiment object are output from DSeq2, HR, Lima, or a simple data frame. We can also add results to an existing DeeDeeExperiment object using the addDea method. Here we add one single object, or we can add a named list of elements. And we can inspect a little bit what's happening in the raw data, and we can see that for each contrast we have we store three elements, 3 main components, which are the log FC, the p-value, and the adjusted p-value. Same for the FEA results. So we can add FEA results using the addFEA method. You can add a named list of contrasts or a single object, or we can add informations like here in this example, the tool that was that generated the results, and we can inspect or like see what kind of results formats that we can use using the support FEA formats function. We can link DEAs and FEAs inside one DeeDeeExperiment object using the link DEA and FEA function or method, where we have to specify the DEA name and the FEAs that we want to link to that DEA. So in this example, we linked 2 FEAs to one DEA. We can also add contextual information to a specific contrast using the add scenario info, where we have to specify the DEA name in question, and we add this information as a string or like free text. And why are we doing this? Well, it's actually this information is meant to help document the experimental setup and clarify the comparisons between the groups. And this information also can provide or help us interpreting the results, since it's a free text, so it can be read by LLM. So it makes sense to have your results and the context of your results all in one place. You can also display a summary of the DeeDeeExperiment object using the summary method, where you can set up the FDR threshold to subset for DE genes. So you can quickly see for each contrast how many up and down regulated genes for a specific FDR threshold, and for the FEA results, you can see for each contrast, for example, the linked DEA, if we have a linked DEA in that object. And we can see the tool that was that generated the results, as well as the number of terms in each contrast. You can also display the scenario info by setting up show scenario info to true inside the summary. And you can get the results table actually with the get data, for example, where you specify the data name that you want to display the table for. And here you can see the minimal view of the results with 3 columns, the log FC, the p-value, and the adjusted p-value. You can also display the results as a list of contrasts. And in a similar fashion, you can use the getFea method to display a specific Fea result, or you can use the getFea list to get all the Feas that are stored in the DeeDeeExperiment object. And if you specify the data name, you get all the Feas that are linked to that specific data. You can rename your DeeDeeExperiment results or the data results that are stored in DeeDeeExperiment object using the rename data. You can rename one element at a time or multiple elements at a time. And same for Fea, you can use rename Fea method. You can remove your results after introducing them using the remove data and similar remove Fea for Feas. So now, for single cell, where we actually generate more tables. For example, in this plot, we have like 19 clusters. So the good news is that you're not gonna, it's not gonna be complicated because you're gonna still perform your analysis as you would normally do, for example, using the MOSClip framework. So you use your PPDS function, as you normally do. You extract your contrast, and what you have to do when using DeeDeeExperiment is you have to rename your columns. The log of C becomes the log2 fold change, the p-val becomes p-value, and p-edge log to p-edge. And then you're good to go. You're good to go to insert these results as a list of contrasts or data frames in this case, directly to DeeDeeExperiment, to create a DeeDeeExperiment object or insert them into an existing DeeDeeExperiment object. So DeeDeeExperiment can be used with Bioconductor tools such as GenTonic and IC, and you can learn more about this in tomorrow's IC workshop. And DeeDeeExperiment is already in Bioconductor. It got added recently to the development version. And with that, I would like to thank you all for your attention. And I would like to acknowledge my group members, the bioinformatics group at the InBioMinds, and also my supervisor, Federico Marini, for their support for this work, as well as the Bioconductor community, especially the people in the developers forum for answering my questions. And that's what I'm going to do now, is answer your questions. Thank you.
Okay, thank you, Najla. Any questions? Yeah. Okay.
So thank you for your wonderful talk. I really enjoyed it. I have a somewhat a bit naive question.
Yes, sure.
I'm not very used to all these methods, the Fea and the DIA methods, but I was wondering, because the single-cell experiment kind of has like this alternate experiment slots, so was it like a problem of dimensions that we cannot fit this? Because if I understand correctly, this DIA and Fea, they give us these matrices of as results, right?
Yeah, it's tables.
Yeah. So was it like a problem of dimension that we cannot maybe store them in alternate experiments? So
no, I mean, technically you can store. The thing is, why are we adding new slots? It's for structure and reproducibility, because you can technically have your results in an Excel file, and then Excel starts to add in dates to your to your tables, and it can be messy. And also, if you have like 18 tables, and imagine that you're scrolling through a folder with many Excel files, or I don't know, even in your R session, it gets confusing, especially if you don't name your stuff the right way. So the solution that we thought about is that you generate your results through your regular pipeline, and then you put them in this container that is structured. So the structure can't be altered. So there are like rules, specific rules that that have to be followed. You cannot put whatever there. So like everything is controlled. And also it is good for reproducibility. So instead of sharing, for example, with your colleagues, a big folder with these tables and whatever, you just share one object, which is the DeeDeeExperiment object, would have the counts, that have the metadata, that have the results, and have context.
Oh, nice. Thank you.
Okay, so let's thank Najla. Yeah, we are out of time. If you want, you can ask her at the poster session. Yeah, okay. Thank you. Okay, so let's welcome our next speaker, Justine Leclerc. She will present conformeR, conformalized differential expression analysis of multi-condition single-cell data.
Hello, everyone. My name is Justine Leclerc. I'm a PhD candidate in Zurich. And today I'm going to present my ongoing project, the building of the package conformeR, that is intended as a new tool for leading differential expression analysis for single-cell, for multi-condition single-cell data. Okay. So usually when you lead such an analysis, you need and prefer methods that control for the false discovery rates, because you deal with tens of thousands of genes measured across tens of thousands of cells. So for multiple testing, it's a nightmare. And then you would like to have some p-values at the cell type level, so aggregated across patients to make inference about larger populations. And last, you would preferably use distribution-free methods so that you can make a few assumptions. So to that, conformeR answers with so bridging conformal inference and counterfactual prediction to output valid results. Okay, so to drive you through conformeR, I would like to present a toy example. So imagine you measure cells of Mr. and Mrs. Smith. So you will only measure two genes, gene A and gene B, across cells of the same type. Randomly, you apply to half of those cells a treatment. To keep things simple, you assume that the log counts of gene A are standard normal distributed, and that the log counts of gene B are just a linear combination with a linear coefficient that changes across condition with some noise. Our research question is gene B differentially expressed under treatment? Basic research question. Okay, so the first step in conformeR is to predict the counterfactual. That is, what would have happened to gene B had we observed this gene under the other condition. So to that, we have several implementations. So either you can just use an existing package. So we have Lima, which is a bioconductor package, or CF causal, or, and it's as well an ongoing project, we have our built-in prediction model using conformal inference. And for that function, we make the new assumption that the condition of one gene, of a gene of interest, can be predicted by the other expressions that we observed on the other genes. So basically, in our toy example, that I can predict the level of gene B by the level of gene A in the same condition. We take genes as playing together. Oops. Okay, so what you do to predict the counterfactual is that you will first learn with the data the conditional distribution of gene B given the level of gene A and the condition in which you observe the genes. So for example, in the toy example we have, we will take advantage of this linear relationship we have on, between gene A and gene B. So for example, if we have no underlying difference, we will have some light treatment effect that is due to noise. And if we have, on the other hand, some different linear coefficient between conditions, we see that there is some smaller or larger difference in potential outcomes depending on the expression of YA. Okay. So once you have the two potential outcomes, you can now go further with testing. So what we do is that, so we test, is gene B differentially expressed under both conditions, so when switching to one to another, and we formulate that as the null hypothesis that the conditional distribution of gene B given gene A is the same regardless of the condition in which we observe him. To test that, we are going to build conformal prediction intervals around delta. So we define delta as the difference in potential outcomes, and we do that over a grid of alpha, over a grid of confidence level. Usually you would basically take a grid between 0 and 1 spaced by 0.01, for example. And then you will just assess, you will compute the test statistic and the related p-value as the coverage of the value 0 across all that grid of confidence interval. Let's have a graphical look. So you see here, so we have for one cell of Mr. Smith, 99 confidence intervals, so for equally spaced alpha between 0 and 1, and we see that when there is no underlying difference, so when tau is equal to 0, we have very few times in which the 0 is covered by the intervals. So you can, I don't know, I don't remember. Yeah, here. Oh. We have it that is, yeah, very few times covered. And on the other hand, if you have an underlying difference, you see that the 0 is very few times covered. So here you have a very small p-value, and here a larger p-value. Okay. And if we look then at the distributions of our p-values across one cell type for Mr. Smith, so here we have 250 cells that were tested. We see that if there is an underlying difference, we have this nice left-skewed distribution of the p-values with smaller p-values for an absolute mean tau that is larger. And on the other hand, if we have no underlying difference, we have this distribution that that is similar to a uniform distribution, and it's what we expect. Okay, so now we have a bunch of p-values. We have technically one p-value per cell and per gene, so it's huge. What can we do with that? And that's the step in which we are going to aggregate those p-values, so as to have one p-value per cell type and per gene. What we do is that since p-values are not that easy to aggregate and to combine, because you may lose lots of precision, sometimes it's not even correct, we will use false discovery rates. A first step is to convert the p-values to local false discovery rates using the Q-value package, and then use the relationship between the Bayesian FDR and its frequentist counterpart, taking the conditional expectation, knowing that the p-value is smaller than an alpha. So now you have one false discovery rate per patient and per gene. Next step is the aggregation, and there we use a previous established result by Brad Efron, that is that you can aggregate the false discovery rate by taking the mean a weighted mean by how much does the patient contribute to the total number of discovery. This is in the case that you have one discovery, because in the case in which there is really like no difference, you may even make no discovery, and in this case you take the normal average. Okay, so we applied it to the case of Mr. and Mrs. Smith, and we're quite happy because we see that when the difference is quite null, so we have larger p-values. When the difference is not null, is not equal to 0, we have smaller ones. So when you're happy with this small example, you want to apply it to a real data set and see if it even works. So we tried with a data set, so with single cell data of 7 COVID-19 patients and 5 healthy controls. We selected only one cell type, the PDCs, and 8 genes. So it was just to see if the workflow even worked. I highlighted because we had different results than the authors of the paper, but it's not contradicting the performance of conformeR, because here we only use 8 genes, so 7 genes to predict the 8th one. So yeah, it's just to see, does it even work? Okay, so the upcoming challenges, because it's a really young project, we would like to submit conformeR to Bioconductor. So for that, we have to finish our internal counterfactual prediction method. We need as well to integrate with Lima, so the package from which I spoke earlier. Optimize speed and computation, because you're treating so much of data. And finally, evaluate it on a larger data set with, so it's a COVID-19 patients data set with 864 patients, so perfect for aggregating across patients. And with that, the take-home messages, so conformeR brings conformal inference to single-cell differential expression. It provides robust p-values under weak assumptions, and it proposes to aggregate cell-level findings across patients. And with that, I thank you for the attention.
Okay, thank you, Justine. Any questions for her? No? Okay, I can ask. Which do you think are the use cases that would benefit the most of this method?
So basically, I think that the advantage of this method is that it clearly takes advantage of the relationship between genes. It's kind of new, because normally in the existing methods, it's one model per gene. And here you really propose to take all the information you gathered to output those p-values. So I think that can make the difference. Yeah.
Okay. Any question? Yeah, okay.
Yes, thank you for your talk. Maybe a very nice statistical question. So you described the procedure to infer the FDR based on the p-values.
Yeah.
Could you just maybe remind me why you don't use classical Benjamin-Lee-Hochberg correction for that?
So the passage to the FDR is just to go from the stage in which you have a bunch of p-values, so 1 per cell, to the stage in which you have 1 per cell type. Benjamin-Hochberg is just to control for the FDR. So we take the FDR because we can't take the p-values that are not that easy to combine. Yeah.
Okay, thank you.
More questions? No? Okay, so let's thank Justine. Thank you. And yeah, let's welcome our next speaker. Wait. Okay. So we have Lizhong Chen, who will present ELIXIR version 4 with expanded functionality and improved support for small counts and larger datasets.
Okay, so Hi, everyone. So it's my pleasure to report some recent updates of the AGR with the expanded functionality and the improved support for the small counts and the large data sets. So at the beginning of this year, we published the AGR-v4 paper in the NAR, which is a review paper which summarizes all the development since the very first beginning of AGR version. And we introduced the new co-selected pipeline in this paper. So the main assumption for the new co-selected pipeline is the mean variance relationship, which is a very simple one. We can separate the variance in two parts, the first order and the second order. And the two parameters represent like the sigma square, which is the co-selected dispersion, which we mainly accounting for the technical of dispersion, and the negative binomial ones, which is the psi, which will account for the biological ones. And traditionally, we estimate the trended dispersion or the tag-wise or the common ones, but for the current, the new co-selected pipeline, we only estimate the global, the BCV, which we only use the highly expressed genes. And a potential assumption for this one is we assume that the gene, there is no specificity for like such gene has a large BCV or smaller one. So, and with the new co-selected pipeline, we can apply that to a single cell RNA-seq data, which is example data, which is a memory grant. It consists of three major ones, LP, BASO, and I think the ML cells. So it has like 9,000, over 9,000 genes and 3,000 cells. And if we run the classical pipeline, the AGR-v3, we need to run the estimated dispersion and the GMQ fit. So you will see it takes a lot of time in the estimate dispersion function. And if you run the AGR-v4, you can just run the GMQ fit, so which is much faster, like 70 seconds over like 300 seconds. And so what's the difference between the AGR-v3 and AGR-v4? So the first one we can see for a typical single cell data, you can see the BCV plot, which has a very left skewed and very high display BCV for the small counts. And it decreases to the large counts and it becomes stable. And if we run the AGR-v3, we can see the co-selected dispersion estimate become very, very biased when the counts are relatively small, which is close to 0. But with the AGR-v4, we update the adjusted deviance status. You can see the co-selected dispersion plot. The co-selected dispersion estimate is very robust. It's very close to 1, which is what we expect for this data. And a simple application for the adjusted deviance statistics is we can use this as a good little fit test to find the highly variable gene, which is very important for the single cell data because we use the highly variable gene for the clustering, for the dimension reduction and the clustering. So what is the high variable gene, which we assume like for the low hypothesis, we assume like all the cells are from the single population. So the variance, we assume the variance, and for simplicity, we assume the sigma square, the technical ones are the same for all the genes, and the biological ones is measured by the psi, and the highly genes, the HVGs are those genes with a relatively large BCV psi G. So what I mean by the relatively large? So it's the 5G larger than the common ones. So in this case, the HVG can be classified in two categories, which is low is accepted, that is the 5G is relatively large, or the low is rejected, because that the expression varies between the cell types and result in a very large BCV. And what AGR does is like AGR count does not estimate the 5G, the BCV, directly, but AGR can estimate the sigma G and the sigma square. So in this case, we can just to test whether the model, the fitting, the low hypothesis is adequate or not. So which is a good fit test using the adjusted deviance. So we can check the results. And we can see from the left figure, the red one, the blue ones, and the green one, which is the top 100, the top 200, the top 400. And you can see the AGR approach prefer to select the highly valuable gene in the highly expressed genes. And we can also compare the result with the CIRAT, for both top 400. And you can see the difference is very clear. CIRAT will pick a lot of highly valuable genes with the expression level is relatively small, but AGR prefer to select HVG, which has high gene expression. And this is not a benchmarking which is better or not. It is just some different choice. And so we can see some selected highly valuable gene. And you can see two are slightly different, which is the force and the gene. You can see the force and the gene, their expression level is relatively high. And you can see the variance in every major cluster is large. You can see the relative from 0 to 6. So what means the force and the gene, they are highly variable in all cells. And interesting, those two genes relative to the cell stress. That means it is a type of the cell state and not related to the cell types. And we can see also other genes, and you can see they are not expressed in all ones. That means the low hypothesis is rejected. They are only expressed in one or two major clusters or the small clusters. So this is a direct application of the AGR for the highly valuable gene selection. And we can also use, and we also update the prior empirical base, which is very important for Lima and AGR. We can, at the very beginning, Smith update using the moment estimate, and the trend is introduced, and the robust one is one. And for the contact holder, we will see the genetic frame is not a constant and varies a lot. Many DGs can be very small, so we update the new functions in the feeder district unequal DF1. And the result is very clear in the transcript simulation. We can see with the similar transcript data, the AGR, the 4.4 can handle the FDR, but the AGR 4.2 fails because the prior estimation is more accurate now. And so because of the improved estimation, we can perform some type of the DE analysis for a single cell there, which direct application is the mark gene selection. Mark genes is very important to identify the clusters usually with the positive log for the change. And the cluster specifically is preferred, but for one gene, one cluster, but it can be a mark gene set which specifies some the transition state. And with the cluster are defined, AGR performs the one versus rest. And for the one sample, we can perform on the single cell data. For multiple samples, we can perform on the pseudo bulk is recommended. And so we construct the contraster, and we run the GM-curve test and perform the DE on the single cell level. And we can see the p-values are not reliable because of intercorrelation among the cells, but we can have the rank is almost useful, and we can identify the top genes like 12, CAS, and the CD14, which is the mark gene for the LP cells. And for this approach, the top D genes may not be classical specific, but it can be a useful set to identify the cell types. For the log-fold change, we prefer a log-fold change for the mark gene. In this case, a treated style is recommended, not a direct log-fold change cutoff. And the last in the AGR 4.6, we introduced the AGR diff splice function. And this is a preprint and will be published soon in the NAR. We can see the AGR diff splice perform the best in the DTU analysis. And so for some feature work, treated style analysis will be introduced similar to the Lima ones. And we will also introduce the sample weights similar as the Lima ones, which to account the sample variability, which is very important for the human data. And also, we will update a new quality pipeline for the methylation data, which is the two count magic data. And lastly, I want to thank Gordon Smith, Professor Gordon Smith, to bring me into the AGR project and other members of the Smith lab, and we has communicated. And lastly, I want to thank the support from the NHMRC funding. So I'm happy to take questions and happy to talk if you are, for further details if you are interested. Thanks.
Thank you. Any questions? Don't be shy, come on. Okay, maybe I can ask something. So I've used AGR to analyze ataxic data.
Yeah.
Do you know or have you tried to use this new version for single-cell ataxic data? It would work?
I'm like for the RNA-seq data, I believe AGR works perfectly on the RNA-seq data. For the ataxic data, I do not have much experience, but in somewhat it works very well in the single-cell ataxic data. But it does not work very well for some type of like high-C data or the methylation data. So because you will see like the coverage for the methylation, the coverage is very short, and you can't identify a significant mean variance relationship, especially for quadratic ones in that data. But if you can see like the BCV plot like this one, so let me just take 1 minute more. So I think, yes. If you can see like a BCV plot, like the left one, it goes from the high to low and becomes stable in the right tail. The AGR will work perfectly in this case. So if you check the BCV plot first. Okay.
Thank you. Any more questions? No? Okay. Well, thank you for your presentation. Okay, so let's welcome our next speaker, which is Koen van den Berge, and he will present transcription factor activity estimation via probabilistic gene expression deconvolution. So this is the laser, and this is to move forward.
Okay, thanks. So I'm Koen van den Berge, and I'll be presenting a project we've been working on to estimate the activity of transcription factors. This is a collaboration with Peter Bickel and Sandrine Dudois from the University of California, Berkeley. So the motivation of this problem, or of this project, is really that we are often confronted with large single-cell RNA-seq datasets comparing different patient cohorts. And one of the end results of these analyses is a long list of differentially expressed genes comparing the two patient groups for each of probably many cell types. In my view, then it's still really, really difficult to interpret this long list of differentially expressed genes. Some people only look at the top. You can do gene set enrichment analysis, or you can summarize these into modules or any other kind of downstream interpretation analysis. So we've been thinking, and of course genes are governed by transcription factors. We can encode these links into gene regulatory networks. And our thinking is really to shift the paradigm from interpretation of differences in gene expression to interpretation of differences in the regulatory effects of the transcription factors that are resulting in the transcription of these genes. So what we're gonna try to do today is that we're gonna define transcription factor activity by assigning observed messenger RNA molecules to the transcription factors that may have produced them. So I'll start with just a brief concept. This is not exactly how the method works in practice, but it should give you a basic idea of what we do. So here we have a gene regulatory network of 2 transcription factors in green and 3 genes in blue. Both transcription factors are regulating gene 2. Gene 1 is only regulated by transcription factor 1. Gene 3 is only regulated by transcription factor 2. For gene 1 and gene 3, it's fairly trivial. We can assign the observed RNA molecules only to one transcription factor. So there's really only one thing we can do there. And we can see that transcription factor 1 produces 9 molecules, transcription factor 2 produces 6 of them. So we make a major assumption, which is by assuming that a transcription factor is equally active across its constituent genes. If we use this ratio of 9/6, we can basically assume that transcription factor 1 is 1.5 times more active than transcription factor 2. We can continue this reasoning by assigning the molecules of gene 2. So this is the same 9:6 ratio. And then we sum all the molecules that have likely been produced by transcription factor 1 and by transcription factor 2. And we personally are defining transcription factor activity as the average number of molecules a transcription factor produces among all the genes it is regulating. So this is really the conceptual idea of the statistical methods. So a bit more formal, think about a single-cell RNA-seq dataset where we have known cell type labels. We have gene regulatory networks with genes in the rows, transcription factors in the columns. For the sake of the presentation, the matrix is binary. The statistical method also allows to incorporate repressing links, so negative ones. And what we're really interested in is this latent random variable ZGTI, which is the unobserved number of molecules of gene G that have been produced by transcription factor T in a particular cell from a cell type C of I. If we let the expected value of this latent variable be equal to mu GTC, then we can note that if we sum these mu GTCs across all the transcription factors that are regulating gene G, then we basically have the expected number of molecules of that gene in the cell type, right? So really what we're trying to do is right, we can easily estimate this using a sample average of all the cells of cell type C. We just average the observed gene expression of that gene. What we're trying to do is that we're trying to deconvolve this into the mu GTCs, into the molecules specifically produced by specific transcription factors regulating each gene. If we have those, we can define the transcription factor activity, again, as the average number of molecules the transcription factor produces among all its constituent genes. We make two main assumptions in our statistical model. First, we assume a multiplicative model for these mu GTCs that only consists of a gene-specific effect and a transcription factor-specific effect. We also have a gene regulatory network there to ensure that there's a link between the G and the T that we're trying to link here. So this is just a very basic non-parametric multiplicative model. Our second assumption is that we assume that this transcription factor-specific component is actually equal to the transcription factor activity that we have defined as before. So these two main assumptions and the definitions we had previously is all we really need to estimate these parameters. So this is a non-parametric model, and we can use a method of moments estimator to estimate these parameters. What we've been doing in the paper is that we've been building on top of that. And we've been developing a few flavors of different statistical models. So the first one, our most basic one, is a frequentist model. And it basically assumes that these latent variables, so the unobserved number of molecules of gene G produced by transcription factor T in cell I, are Poisson distributed. And this allows us to fit our parameters using an EM algorithm. In a second version, we extend upon this by allowing you to incorporate prior information on the gene regulatory network, right? Sometimes you have kind of confidence scores for each edge in the gene regulatory network. You can incorporate that. In this case, we do this via Dirichlet on the relative contributions of transcription factors to genes. And this has the additional advantage that it allows you to borrow information across cell types to estimate these pi GTCs. As a last option, so suppose you would like to take advantage of the borrowing of information across cell types, but you don't have prior information. We also allow you to empirically estimate these prior parameters in every iteration of the EM algorithm in a kind of empirical based fashion. So we fit all of these flavors using a pretty standard EM algorithm. So I'm going to skip over the simulations. I'm just going to walk you through a few very basic results on a case study. This is a data set on the olfactory epithelium that picks up smell signals in our nose and sends them to the brain. What we've been doing is that we have been injuring the olfactory epithelium, and then it basically regenerates the neurons that pick up these smell signals. So in this tissue, horizontal basal cells are stem cells, and they develop to produce mature olfactory sensory neurons. This is a trajectory. We fit a trajectory through this. We calculate pseudotime for every single cell. It's a latent variable, as Susan has been saying, really telling you how far each cell is along this developmental process. We use the pseudotime to classify cells into 20 different bins. Think about these as cell type-like. We estimate the gene regulatory network based on the data, and then we use that to estimate transcription factor activity using our statistical method. This is a very basic visualization of the results. So in the rows, we have 235 transcription factors. The columns are these 20 different pseudotime bins, stem cells in the left, neurons on the right. The pseudocolors of the heat map are representative for the estimated transcription factor activity in each of those 20 different pseudotime bins. So the transcription factors in the rows basically cluster into 3 different groups. Those that are most active at the stem cell stage, and those are then also typically involved in gene sets relating to tissue development. You can see cellular response to chemical stimulus. We've performed a chemical injury to the olfactory epithelium. In the middle of this trajectory, we've got the cell cycle, and you can see the gene sets popping up there. And then at the end, we've got the neurogenesis and some gene sets related to that. My favorite visualization is the most basic one. So here again, the 20 pseudotime bins on the x-axis. Every data point is a single cell. On the y-axis here is the relative contribution of a specific transcription factor. So we've got 4 transcription factors here. Because we deconvolve the observed RNA molecules to transcription factors producing them, we can look at measures which we're calling relative transcription factor contribution, which is really out of all molecules produced, what's the fraction produced by this specific transcription factor? You've got HDAC2, which is more active as neurogenesis occurs. This is indeed a transcription factor responsible for neurogenesis. RFX3, similar pattern. This is responsible for cilia formation in neurogenesis. FOS, it's been mentioned before today. This is responsible for differentiation and activation of stem cells, and we can see indeed it's mostly active at the early stages of the trajectory. So our model allows transcription factor activity estimation using a probabilistic model. To the best of my knowledge, but feel free to let me know if you think otherwise, this is the first method that really attempts to probabilistically deconvolve gene expression by assigning RNA molecules to the transcription factors, leading, therefore, to interpretable transcription factor activity estimates. There are several features, such as sparse results, repressions, and a specific distance-based ranking of transcription factors that I have not touched upon, but we have a preprint. And I'd like to finish off with the limitations of the work. So the method obviously relies on a known gene regulatory network. If this network is of low quality, the results will equally be of low quality, right? Your network needs to be good enough. The assumptions of the model, especially the fact that we assume that a transcription factor is equally active among its regulating genes, is unrealistic and are restrictive. You've got cooperation and binding motif preferences, but nevertheless, it's our hope and our belief that the results are still useful. So I'd like to thank Peter Bickel and Sandrine Dudois, my 2 main mentors on this project. We have a preprint on bioRxiv. The software is currently on GitHub, and we're preparing it for Bioconductor. Thanks.
Okay, any questions for Koen? Yeah.
Hi, thank you for your talk. Apologies if I missed this detail, if you actually went through it. But the effect of transcription factor can be bidirectional on the gene expression, right? So do you account for that, or do you just assume that transcription factors always just leads to upregulation of a gene?
No, so we do account for repressions also, where transcription factors basically downregulate the gene.
Mm-hmm.
There are specific extensions to the statistical model that we've implemented to do that. I just didn't go through them in this talk. Basically, we model what we think the effect could be, and then we try and subtract for it. But I'm happy to provide more detail if you like.
Also, another question. Does your tool take in an input of the annotation, which links the transcription factors to the genes?
Yeah, so we require a gene regulatory network as input.
Okay, and maybe we can just talk about it later. And that's probably where you also input the associations, right? Negative and positive associations.
Right, exactly.
Okay, yeah.
Okay, any other questions? Yeah.
Thank you very much for your talk. I had a question regarding the direct effect of transcription factor to the gene expression. Have you ever thought about also analyzing if there is any direct relationship between, instead of the gene expression, the genetic variants? For example, some method like Mendelian randomization to understand if it's exactly the causal of a manifestation of a phenotype directly from a genetic variant, or is there is a transcriptional factor that is a causal in this case?
So we did not really think about genetic variants in this case, nor about causative agents or causal inference. To us, this is really a method for interpretation. Really, the main output is just ranking transcription factors, seeing which ones are more involved or less involved in the project as compared to others. But in terms of genetic variants and their effects on the transcription factor activity is not something we've been thinking about. Thanks, though.
Okay, so let's thank Koen. Okay, and our next speaker is Anna Tanada, and she will present integration of multi-omics data with topological pathway analysis tool, case studies with MOSClip.
So good afternoon, everyone. So thank you for the opportunity of being here and talk a little bit more about our package MOSClip. As you can read from the title, I'm going to be presenting some case studies to understand better the applications of this tool. So starting from the basics, what is MOSClip, right? The main functionality of this package is to perform topological pathway analysis of multi-omics data. The idea of MOSClip was born from the difficulty of integrating multi-omics data and especially understanding the impact of each omic and their combinations on biological processes. So this package actually was previously published on 2019, but we have upgraded and updated, and it's been on Bioconductor since last year. So in the next few slides, I'm going to show a little bit about these novelties of MOSClip. So also, I'm going to explain very briefly, the method because, time constrictions. But you're welcome to visit my colleague's poster today if you want to know deeper, the details of how MOSClip works behind the scenes. So MOSClip can perform the analysis in two different levels, pathways or modules. Pathways is just you take a list of pathways from the database of your choosing, like also in all these case studies, we're using Reactome. So you take all the genes from the pathways, or you can use the modules level. The modules, we basically decompose these pathways into these gene modules, using graph theory methods, like moralization, triangulation,
etc. So we take the genes from these pathways or modules and extract them from all the omics the user put as input, and we perform a multidimensionality reduction step that is omics specific. So we have several methods too. Once we perform this step, the user can perform the analysis per se. And we have two types of analysis. Originally, MOSClip was created thinking only on survival analysis. But now, as I said, we upgraded and we can also perform a two-class analysis. So basically comparing two different conditions. Also, MOSClip have multiple plot functions integrated to help the user visualize the results. I won't have time also to show all the plots, but hopefully you'll see the cuter ones here on my study cases. First of all, the first case, of course. Here, we want to show two novelties of MOSClip, which is the two-class analysis and also a toxic data that before was not compatible. For this case, we're using breast cancer data from the Cancer Genome Atlas. The omics here used, as I said, ATAC-seq, copy number variation RNA-seq from 46 patients. The comparison here is Lumino A versus Lumino B patients. A quick context for those that maybe are not very familiar with breast cancer. Lumino A and B are molecular subtypes of the breast cancers. Of course, it's not the only two molecular subtypes, but it's the one we are using here. So as you can see in this table, they have some differences. One, for example, is that Lumino B usually has worse survival and also have higher proliferation. And I'm going to go back to this point in the future. This is the first plot we have. This is also a result that MOSClip can provide the user, sort of a heat map summarizing all the results. In this case, we're looking at the top 20 most significant pathways that also are significant for at least two omics. So the first column, we have the general p-value of the pathways coming from the model. The other columns are the p-value of the covariates, in this case, the omics. So here in red is ATAC-seq, and if you're wondering why we have 3 columns for ATAC-seq, it's because we performed the multidimensionality reduction step, and here we are showing the p-value for the first 3 principal components of the ATAC-seq. Then in green, we have copy number, and in blue, we have expression data. So this is a very nice way to quickly visualize the results. So for example, if you don't have any pathway in mind, just looking quickly on this figure, we see these 2 pathways. They are related to cell cycle and probably cell proliferation. They are probably very interesting to take a deeper look, since I said that Lumino B presents a higher proliferation. Another plot and test that MOSClip can perform is the super-exact test. The exact test is basically a computation of the statistical distribution of the omics combination, and then you have this circular plot. In other words, MOSClip is going to take a look in the number of the pathways or modules. In this case, specifically, we're looking for modules of pathways. So the number of modules they have for each omic combination. Let's look closely to this part of the graph. So each ring here is an omic, and the outermost ring is the p-value for this omic combination. If it's yellow, it means that you have this omic present. So, for example, we have 139 modules. They are apparently highly significant for the omic combination ATAC-seq and expression. Another novelty that will be soon released is that MOSClip can help the user to plot the results on Cytoscape. So MOSClip will allow the user to help creating the data frame with node and edge attributes, and also all this design, and also reducing the redundancy of the results, because as you might know, a lot of genes are present in more than one pathway. So these functions will allow to reduce this redundancy and plot the results in a visual way. Here are illustrated the significant pathways that are significant for the combination ATAC-seq and RNA-seq. ATAC is in purple and expression on red. Now, the second case study, which here we're going to represent the most classical MOSClip analysis, that is the survival mode, using ovarian cancer data from the TCGA. The omics used here are expression, copy number, methylation, and mutation from 262 patients. This is another plot that MOSClip can do. This is a frequency distribution plot as a radar plot. So basically here it shows the frequency of pathways that falls under these macro categories. So breaking this down a little bit, we see there is a peak for the macro category immune system for the omics combination, copy number, and expression. And then in this way, you can quickly also summarize and see where all the pathways are falling under these macro categories for each omic combination. Since we're talking about survival, of course, MOSClip also plots Kaplan-Meier curves. And we can plot these Kaplan-Meier curves for specific pathways or modules. In this case, we are representing the module 2 of the pathway interleukin-12 family signaling. And then it plots all the possible combinations of the covariates. So in this example, we see we have a significant p-value, and this light green curve seems to have a better prognosis. This light green curve is the covariates represented are here. So it's, for example, for few negative copy number, low values of the principal component 1 for the expression, and high values of principal component 2. So in this way, the user can understand in deep detail of the module exactly how the omics are impacting survival. Now, just to conclude, since we talk about a lot of things, maybe you already lost midway of the presentation. So MOSClip is a tool to perform topological pathways analysis from multi-omics data, and is publicly available on Bioconductor now. When you use MOSClip, keep in mind that you can perform 2 types of analysis, survival or 2-class. And within each analysis, you can also perform in 2 different levels, pathways or modules. Currently, MOSClip is compatible with the omics RNA-seq, attack-seq, mutation, copy number, methylation. We also have multiple plot functions integrated to help visualize the results, and so we'll also provide tools to help plotting with cytoscape. And as future perspectives, we are thinking about amplifying the omics that are currently accepted, and we are working on single-cell data. So I would like to thank to everyone that worked in this project, and also special thanks to all my lab mates and colleagues, and everyone that is here present listening to me. Thank you.
Okay, thank you, Anna. Any questions for her? Yeah.
Hi, thanks. Maybe I missed it, but in which format do you expect this multi-omics data to be in? Multi-assay experiment or something different?
Yeah, we are also compatible with multi-assay experiment, but if you also have like a list of all the matrix, then you can perform, you do inside the object, there's a list, and then you put all the omics. But like, yeah, we basically use a multi-assay experiment object inside our object. I don't know if it was clear.
Yes, okay. Thanks.
No problem.
Any other question? Maybe I can ask one. Yeah. So you say you use attack-seq data in some instances. How do you link these regions to the pathways? Do you use only promoters or by?
Yeah. So to calculate in the model, we put the peaks. But then to annotate them to the pathways and to the genes, we are using ChIP-seq. Like this should be a pre-processing step that the user should do by themselves. And then once you have like the annotation, you also put inside this object, and then MOSClip can do the rest.
Okay, so you can provide the annotation yourself if you have another way of annotating or whatever?
Yes, exactly. If you have another way, it's just basically a data frame, and then you use as input.
Okay, nice. Okay. More questions? No? Okay, so let's thank Anna. Okay, so let's welcome our next speaker, which is Igor Cervenka, and he will present Rega, our interface for metadata submission to the European Genome-Phenome archive.
Hi, I'm Igor, and I work in a bioinformatics core facility in the University of Basel, and I would like to thank the organizers for the invitation and to give the opportunity to present some of our work. And the talk might be a little bit different. It's about a third-party client that, with any luck, would make some of the lives easier. If you work with human data in Europe, that is, I apologize to our intercontinental colleagues, then you probably heard of EGA, which is a European Genome-Phenome archive, which is one of the solutions for archiving unidentifiable and sensitive human data. In order to submit your data set to EGA, your institution needs data access committee, which will see that if they allow you to submit data to such control repository, and you also need somebody with an approved submitter role. And unlucky for us, in our department, we are the only ones with this role, so we have to handle all the submissions. And also, unfortunately for us, the submissions to EGA is not as straightforward as to GEO. It contains a lot of different metadata fields and objects with all sorts of weird relationships. And also, they have some sort of a web tool which is somewhat functional. It tends to bug a lot. And, but also for a larger data set, some of the ones that we have around 1,000 FASTQ files. This becomes unwieldy very quickly. And and the whole process in between the approval, the submission, and the back and forth with EGA, this process can take around several months. So out of this necessity, a vision was born to create a package to streamline these submissions. And we had several goals in mind: to improve communication and metadata sharing with the researchers, and also kind of reduce the back and forth with the EGA to validate as much as we can before we submit. Originally intended as an internal package, then we decided to open it up so people struggling with similar things could benefit as well. And we divided the package into 5 sub-modules. They are in quotations because they are mostly conceptual groupings rather than strict modules. And we also provide an Excel template-based metadata, Excel template for the researchers to fill in, and also an R client built in using the EGA specification, which can, in theory, this is rather flexible, so they can be also replaced by a custom solutions. And why we chose the Excel metadata template, since researchers are familiar with this tool, they are also familiar with the GEO submission, some of them, and works on majority of operating systems. It's rather simple to share it, and it has some of the useful features for us, which are drop-down menus, for example. However, the EGA accepts JSON, which can include arbitrarily nested structures, arrays, and whatnot. And also, there are a lot of many-to-many relationships in their database, and also all sorts of enums that they use with specific IDs. I will just quickly guide you through how we decided to structure the package. So first one is this kind of a parser submodule, which transforms the Excel metadata into our object that can be fed into the API. It's mostly a lot of data wrangling, joining nested structures. We try to kind of model their database in an Excel in some sort of normal form, which ended up in 13 worksheets, fillable worksheets. So then we have to deal back and kind of organize it into the data that they accept. This is not super exciting, still important. The next one is validation submodule. One of the problems with the EGA is that it uses their internal accession IDs, which are only issued once the objects are entered into the database. And we still have to somehow track the internal relationships in the dataset, so we use sort of temporary IDs. Then they get replaced once they become available. And we also use a very nice rvalidate package to check if these temporary IDs are present, unique, exhaustive. We also have some additional checks that are imposed by EGA, and the new rules can be added as needed, or new validators can be created as needed. And then there's kind of a few small authentication functions that just try to avoid that you store your keys in an unencrypted form, and also to provide an access to the EGA either via an OAuth or by using the bearer token explicitly. And this was kind of the most fun thing to do. The EGA contains 131 API endpoints, and there are separate sets for data sets that are accepted versus not yet accepted. And this was quite annoying, so how we tried to approach this was to sprinkle it with some R metaprogramming that take care of construction of the endpoint function. So basically, there is an API YAML file that you can download from the database. We parse it, we create the kind of function formals from the required and optional parameters, and then we basically populate the body with the diffused expressions that can handle URL substitution, payload validation, authentication, and creating the request and parsing the responses. In theory, this implementation can be swapped for some other one, like some CodeGen client, but from my experience, the R-based CodeGen clients are terrible, and I have never found any that worked. And also, I actually realized that with a bit of tweaking, you can use this to process any YAML API YAML file to create an R client to any database, basically. I have not tried that, this is a speculation, but it in theory should work. As I mentioned before, we also have some diffused expressions for payload validations in the function, and since some of the newer versions of the Open API schema has some of the more complicated relationships, like one-off, all-off, we would not want to do this manually, so we use kind of a similar approach that we parse the YAML API file, scan for required attributes, and then use the JSON validate R package to basically inject this as a diffused expression. So we make sure that the required types and the values are present in your data. And then the last is the workflows module, which is where all the magic happens, but under the hood, it's basically a series of API endpoint calls with an interspersed data processing. So for example, for a new submission, it asks for the files, submits the samples, retrieves all the accession IDs, replaces it in the data set everywhere that they are needed, submits another object, let's say an analysis object or a run object, and then basically rinse and repeat. We also built in some nice functionalities or functionalities that we think they are nice. It can log information. It can also delete incomplete entries upon errors. And users can also create custom workflows using the API submodule if needed. And how to use it, you navigate to a GitHub repo, you clone the repo, you take the provided Excel template, you fill it in, you can parse it, validate it, you can create the API client, and then create a new submission with your data in an API client, optionally logging the responses, or use some of the other workflows that we provide. Of course, not everything is a walk in the park, and there are some issues and rough edges. Until recently, there was no test environment that the EGA provided, so I had to test everything in their production. So hopefully they use at least 16-bit ints for their primary keys. And Some of the more complicated workflows are missing. For example, publishing already, updating already published data, since we didn't have a real use case for this from people from our department, and I could not really test it against somebody else's data set. And we are still discovering new hidden constraints that are imposed by EGA API. So this is still a work in progress. And I would like to thank you for the attention, and also the Bioinformatics Core facility for their amazing help. And I will take questions.
Thank you, Igor. Any questions? Yeah.
Thanks. I mean, I think only the people who have gone through the pain of submitting something to Rega know how much value is this, how much valuable is this, can be this work. One question, how is it if I understand this is done independently from EGA, it's out of your own core facility to facilitate the submission, but how do you plan to update the package if the EGA API changes? Would it make sense to talk to the EGA people and try to, have a coordination between this development and what they do?
Yes, exactly. That would be ideal. We try sometimes, since I think they don't have that much stuff, so the responses are not always easy to get, as maybe some of you know. In theory, we have their API, the YAML API is bundled with the package, so every time you create, it reads the YAML file and creates the API. So if you would substitute it by a new one, it would basically reflect the new endpoints. It's just then the workflows would need to be modified. The API, some modules should deal with everything fine. The functions constructed will be fine, but then the workflows would need to be modified if some of the tables are named differently. But I think that is not the worst problem, because now they are creating all these federated EGA nodes across different countries. And hopefully they will all not have all different APIs, because then we would have to keep a list of basically endpoint functions for every country. So let's hope this will somehow get solved.
Any more questions?
Thank you for your talk. I was wondering, I have never maybe seen the documentation of the REST API of EGA. I was wondering if is publicly available, or you should register or have an account to access the API.
So the API specification is publicly available as a YAML file from their site. Unfortunately, there are some other constraints that they have on the metadata that you submit that you only get as an error when you try something. So, I don't know, you are trying to submit, a variant data set, and you did not specify a genome build, and then you will get an error. But if you try to submit another type of data set and you specify the genome build, you will get an error because it should not be there. And this is not declared in their API because it only deals mostly with the required fields and the data types, but not their individual relationship. So this is kind of the last point that we had the most problems for, and we still discover new ones even after a few months of development and trying to submit the data sets. So.
The last question that I was having also is this R package is providing any other structure that is different from what I would, if I queried the API directly without our interface. Do you have any other additional processes that you added that will help maybe the person to do further analysis, or
not for the analysis. The only structures that it basically uses is kind of data frames to return the data. And I think this was one of the biggest problems trying to publish this package on Bioconductor, since we don't depend on any existing bioconductor package. It's not necessary. It mostly uses JSON and HTTR2. So yeah, not as it is.
Thank you.
Okay, perfect. So let's thank Igor. Okay, so let's welcome our last speaker of the session, which is Katharina Heil, and she will present ELIXIR for the bioconductor community, resources, networks, and opportunities.
Thank you very much. Yeah, my presentation in the next 10 minutes will be far less technical compared to whatever you have heard in the last hour or so. It's really nice to be here and meet many of you. I think there's very few that I have coincided with before. My name is Katharina Heil. I'm the program manager for communities and training at ELIXIR, the European Research Infrastructure for Life Sciences. So what am I actually talking about? Maybe this one? Yes. So just a couple of words on what ELIXIR is and how we work. So as I said, ELIXIR is a research infrastructure. Legally, we are an intergovernmental organization bringing together life science resources. What resources are we talking about? These are databases, training, software tools, data standards, compute resources, but also scientific and technical experts. So we really want to bring together and raise capacity across Europe for researchers to be used within Europe and beyond. At the moment, we have 22 member states and 3 observers, and we are coordinated by the ELIXIR Hub, and that's where I work. We're generally based in Cambridge. I personally spend quite a lot of time here in Barcelona as well. And ELIXIR works with 5-year scientific programs. What you can see here are the 4 pillars or the 4 tiers that we're working on currently. They are ranging around science, technology, nodes, and the people. And what you can see to the right-hand side here is the very basic underlying structure of the research infrastructure. We work with 5 platforms. They develop and supply and maintain the technology. And at the moment, we have 18 scientific and technical communities. That's what you can see in the outer circle here. You can't really read it. No, and it's not on the next slide either. But they really range from very different technicalities and expertises. So some of them, or one of them, looks at IDPs, intrinsically disordered proteins. We have a galaxy community, which is very technical. We also have a plant sciences community. So they don't do everything that has to do with plants, but they choose what is strategic for them at a given point in the area of plants, for example. What I wanted to bring to this audience is also a little bit of an insight into what are the services ELIXIR brings to our researchers. So what you can see here, the services are run by our nodes. That's the 22 countries that are parts or members of ELIXIR. And currently, the number is probably not correct because it varies probably every other month a little bit. But we have about 600 services for life scientists and bioinformaticians. So what you can see here, really, this is about compute services, data resources. We're looking at standardization, metadata and vocabularies, accessibility, but also benchmarking for software, tools and confirming information standards. But there's also aspects around training services, verifying training, and so on and so forth. Is it me? Maybe, there you go. But really, what I've been talking about at the moment is all very, very technical. But in ELIXIR, we've realized very early on, it's really about the people that make the community. All of our tools are open, they're all fair, at least that's what they aim to be. So really, we as an infrastructure, we don't gain with the services and the tools that we offer, and it really comes down to the individuals that make this whole infrastructure function. So it's all about together, we connect national infrastructures to mobilize data for the life sciences in the European research area, and very often refer to ELIXIR as a people infrastructure as well. So I came here, Maria invited me to talk a little bit about ELIXIR communities as well. I've been broadening it up in the last slides to also really talk about how this could connect to Bioconductor and your community here. So just a couple of words on the ELIXIR communities. I've already alluded to the point that they're very diverse in the topics that they're addressing. In principle and more technically, they're formed around domain experts in ELIXIR nodes. They can include non-ELIXIR partners as well. They provide a mechanism for long-term collaboration with other initiatives, but also other S3s on the S3 roadmap and large-scale initiatives. In principle and in practice, they drive the service development in the ELIXIR platforms, so that's more the technical infrastructure that we're building, and they provide a framework to develop and maintain community standards. If you're interested to read a little bit more about how all this works, some of the principles that we use, some of the incentives as well that we have for people to become or to belong to a community, there's a handbook available on F1000, which you're more than welcome to have a look at. And apart from what I've just said, really in ELIXIR and with our communities, we enable ELIXIR to capture the user needs into formal requirements. We support collaborative standard development. We foster collaboration between the nodes via transnational expert networks, and we facilitate consortium building for funding applications. So we also really look outward. It's not only about ELIXIR and our members, it's about the broad European global community. There you go. So our community communications are bidirectional. So we've got the technical aspects and we have the more scientific aspects. So as you can see again in this circle, which is a little bit more visible than the ones before, we really do want the experts in the field, potentially the end users, but also the people that are still developing tools, to talk to the experts in, say, the actual tool development who don't really know much about the area where these tools could be applied or could be needed, or the standards and so on. So we also really aim for cross-dissemination between our communities. If one community has established a standard which has become the standard to be used in Europe for, say, example, plant genomics, and there's another community that wants to do something similar, we really incentivize them to talk to each other, and that's really also part of my job and my role to make those connections and leverage those. So in principle, our ELIXIR communities are for ELIXIR members, but as I said before, and this is also an open invitation I will come back to later, they're also open to non-ELIXIR people, which are really individuals potentially like you that have an interest in that topic and that can contribute knowledge, expertise, and energy to those topics to really, again, also learn from each other and work together. And this also, again, is to ensure a close link to users and experts in the field. So what are the other activities that might be of interest to you in this room? I've picked up very few and have a couple of slides on those. So one of the things ELIXIR is very strong at is the data management and its resources. So 3 of the kind of highlights and the top tools I would want to mention, and I can add the links to those. They might actually also be hyperlinked on top of those boxes in the slide deck, are the RDM kit. It's an overview of good data management practice, but it really also gives a really nice introduction and a hands-on guide into how to do research data management in your own domain-specific area. The FAIR cookbook includes step-by-step instructions. It contains these recipes to accomplish specific data management tasks and to make data fair. And last but not least, the data management plan wizard is a tool that helps you and guides you through how to write a data management plan itself, again, depending on the needs, potentially adjusted to the domain that you're looking after. I mentioned training before, and I've got this whole slide on this also because it's part of my responsibilities at the ELIXIR Hub. What you can see here is the training life cycle. Again, this is a tool or an interface that has been developed. The hyperlink is at the very left of the slide here. And it talks about the training life cycle. Training is not only about delivering a training. Again, we're also applying the FAIR principles to training. And here we're ranging from designing learning materials, learning pathways, delivering courses, but also impact assessment, and then the full circle to the feedback and potentially delivering a course a second time. One of the things we also do is we bring together people as part of our biohackathons. It's an annual event with about 180 people face-to-face and many more online. We work on 30 to 35 different projects. They're all community-driven, and it's a week full of people coming together, developing and doing something which leads to a result either at the end of the week or further down the line. This is also open for non-ELIXIR members. I know some of you might have potentially even attended one of those. So here's an invitation, no longer for this year, November, because we're all fully subscribed. You could join online this year, but then there's another edition the year after. And with this, based on a conversation I had earlier with some of you, I have added one more slide here around our focus groups. They're a little bit less formal than our communities, but they're really also a very nice entry point for people with an interest in the same topic to come together and to start working on a concrete topic. They're very time limited, but they bring together interested parties or people around a particular topic, as I said. They address an emerging area of interest within ELIXIR, or say the life sciences across Europe. They have a clearly defined goal, and some, but not all of them, will become communities. This is just an example of the ones that are currently active. As they are being reviewed, we have started to close some of them, but there's some new ones coming, which are not on that slide yet. So for me to wrap this up, this is about how I think we can find overlaps and use existing opportunities, but also create new ones between Bioconductor and ELIXIR. As I said, our communities are open to people with an interest and with knowledge, and I think all of you have lots of knowledge and potentially some interest, so you're more than welcome to join our communities. But one could even think about considering setting up a focus group. We have a Galaxy focus group, so potentially a Bioconductor focus group could be something that is also helpful. Join our biohackathon, propose a project, co-lead one of these proposals, and then also just a little bit of a call of action. Let's all work on reusable and fair training materials, add your materials to the relevant repositories, and or join or use the RDM activities and materials that are available. With this, yeah, just one more sentence that resonates with me. If you want to go fast, go alone. But if you want to go far, go together. Thank you very much, and happy to take any questions.
Thank you, Katharina. Does anyone have any question? Yeah.
Great talk. It's really nice to be looped in with all of this. I was just curious, you really kind of focused on, at least you sounded proud about the training material slide. I was wondering how, like your fourth step or something was measuring impact of the training. How do you measure or quantify how well training's gone?
Yeah, what you can see here, thanks for the question, is our training metrics database. So we do have a database that is available for all of our ELIXIR nodes to put in statistics on their training. There's a set of predefined questions that we recommend all our trainers to be running as a questionnaire after their trainings to then really feed back into this. There's short-term and long-term feedback. We have more data on the short-term compared to the long-term. But yes, I think, again, ELIXIR puts in the infrastructure, which is the metrics database, and then it comes to the people to actually feed it with the data.
Okay, any other question? Yeah.
Thanks so much. I wondered about the concept of open source and voluntary effort that is a big part of Bioconductor, and how ELIXIR might be thinking about, is there sufficient investment in something that is open and highly voluntary? How would you contemplate that and present ways of strategizing how to organize your voluntary versus your professional work?
It's a very good question. Let me take a step back, and I think I would say that a lot of our community work has a big component of voluntary engagement of people. We do fund our communities, but very minimally. And I think it's that combination that we have learned, or we've seen that that's what works. The incentives to bring people together, but also encourage them to think about how they can potentially make even a bigger use of say, national funding or project funding. So I think what ELIXIR tries to do in the community space is put the additional clue on top of what is already being funded to leverage and to make even more out of what's already happening. We can have a chat about how to keep volunteers engaged. I think there's a lot about recognition. We do work on just how to recognize trainings, people, software, everything basically. And I think if that improves, then it at least gives, it gives some more formal recognition to it. And I think the big network that we're working on really also has the aim of enabling people to get access to the bigger pots of money. So we're talking European budgets and so on.
Okay, so let's thank Katharina